more later. Tell them they can't eat them yet.
It's not, it's for the presentation, so don't eat it.
Not yet, anyway.
It's part of the presentation.
It's prop for the presentation.
Holding on to this Babylonian system is dragging the world back into the past. Yeah. 666. And now we got Chelsea.
It's part of the presentation.
Good morning. Good afternoon. I'm so on it, I actually remembered to turn my phone to silent. How about that? I just have to brag on myself for a second.
Thank you for the reminder.
But welcome to ASF's monthly meeting. Arkansas Society of Freethinkers is a nonprofit devoted to the separation of church and state, advocating a scientific and humanistic worldview, and improving the lives of non-believers. You can learn more about our organization at arfreethinkers.org, or by talking to the people around you, or through a flyer that we have. We are a diverse organization with members who represent a wide range of opinions. Because ASF is a 501c3 organization, we cannot directly support candidates for public office, but we can advocate for causes such as the separation of church and state and a scientific worldview. Today we're going to be learning a bit about scientific literacy and figure out why we don't have tails, I think. But I'll leave that to the main speaker. Today's free thought of the day comes to us from Brad Bradley, who will also be delivering our skeptical sidebar. And then our main speaker today will be Brad Ericson. So let's give a warm welcome to Brad Bradley.
Good afternoon. The free thought of the day is something that I learned last week. The runtime for my CD version of the album Dark Side of the Moon by Pink Floyd is 40 minutes and 52 seconds. I think it's amazing that the amount of radio blackout that happened when Artemis II went behind the moon was 49 minutes. It was 40 minutes and 52 seconds. We say there's not a God, but it obviously is David Gilmour. So there you go. For a skeptical sidebar, rather than putting something up and then proving it wrong, I wanted to talk to you about something that I read last week. A woman was discussing when does life begin? And when we all started dealing with women's rights to choose and all this, the far right came up with the idea life begins at conception. Anytime you interrupt the conception, you're committing a murder. She said that absolutely is not true, that the truth is life has begun long before that, and you are much older than you think you are. It is known that female humans are born with all the egg cells they will ever have. That's a fact. It is a fact that at least 1 week before they are born, they have all those egg cells. For some reason, there's some debate as to how far back that goes. But you started out life as a fetus, as a fertilized egg. That egg was living and a living being inside your mother. You were existing as an egg cell inside your mother at least 1 week before she was born. I think that's an amazing thing. And her contention is life does not begin at conception. Life began one time on the planet and has never stopped since then. Thank you.
It's a membrane. It's not really a shell. It's like a chicken. It's not a horse.
Cell.
S-H-E-L.
No, C-E-L-L.
Oh, cell.
Cell.
Yeah.
Cell.
Bad mind. Never mind.
The member story is a chance for us to get caught up with one another and to share something with the group that we've been up to, that we've experienced, that we saw. And today's member story, I'm expecting something a little bit triumphant because Jerry Schultz has been working very hard on a particular project for a couple of years that he's going to be telling you about. And no, it wasn't a couple of years, it was several years. So everyone, let's give a warm welcome to Jerry Schultz.
Yeah, it should be somewhat triumphant because basically the message is you won. The monument case, the 10 Commandments, or 11 Commandments as I call it, that monument case has now been decided by the district judge, and we won on every point. And I was involved in all of it. Actually, we had great help from the Freedom from Religion Foundation and from the American Humanist Association in doing the legal work. I didn't do it alone, although I ended up doing a lot of it. And but here's basically what happened to get you caught up. This has been going on for a real long time, because in 2015, the legislature passed a 10 Commandments Monument Display Act, which said we're going to put a representation of the 10 Commandments on the state capitol grounds. And so several other people, including the Satanic Temple, said, well, we want to put up our monuments too. There was a Baphomet from them. There was a suggestion of a Ganesh. They never went through with it. And also the Gold Star families, which is a group representing the survivors of war, people killed in war. They wanted to put one up too. And incidentally, that one is, that one's coming. But here's what the problem was. The way the law was set up at the time, anybody could propose something like this, and so that's why all of these people could propose it. The Capitol Arts and Grounds Commission was required to hold hearings. And I remember I attended, I don't think all of them, I attended all but one, and at that first hearing, before it ever started, they said they had somebody swear in. And swear in, I mean, a new member, you promise, among other things, to support and defend the Constitution of the United States and the state of Arkansas. That will become important later. At the first hearing, it was also interesting that while the TST was there, they did a conspicuous pledge of allegiance, putting, special emphasis on a couple of words. Now, I identify with about 98% of the pledge and skip the other two words. But the interesting thing was, at later meetings when the TST wasn't there, they didn't bother with it. I thought, that's interesting. Anyway, we're on number 3, I think, hearing number 3, and Senator Rapert, the prime mover behind this act, came and basically told the commission, you've got to put this monument there, that's the law. This hearing has all been about whether we should do it or not. Well, that's not your question. It's just, you work out the details. And then Jim back there was at that meeting. A lot of us were at those meetings. There's Jim. And he was the one that was speaking at the time, got to ask, well, and what's the point of this hearing if you've got to do it anyway? And basically, the commissioner says, well, we have to have a public hearing before we do anything like this. And Jim says, but you're not making a decision. And so what are we doing? And the guy said, I don't really know what we're doing here. I don't know why we're here. I don't know why we're doing this. And so I took another turn, and I told the commissioners at that time, it's reported in the Arkansas Democrat Gazette, and then I found the transcript yesterday, but basically what I said is every one of you took an oath to uphold and defend the Constitution of the state of Arkansas and the United States of America. That's your obligation. You're being asked to perform an act that violates the Constitution of the state of Arkansas and the Constitution of the United States of America. Your oath requires you to refuse to do that. That convinced exactly nobody. But anyway, they decided where to put it, set it up June 27th, 2017. And then the next day, a guy crashed it. Actually, I think it was after midnight, the first day it was up. A guy crashed his car into the monument and destroyed it. And there was a lot of jokes about somebody breaking all 10 commandments at the same time. But, and he was not a member of this group, by the way. We had nothing to do with that. So anyway, they had to redo it. And this time, new and improved monument with protective bollards around it so you couldn't drive a car into it was installed. Now, there were a number of different groups of people who objected to this. And it basically formed down into three groups for the purposes of the lawsuit. The ACLU and 4 members of the ACLU, that was one group. This group and the Freedom from Religion Foundation and the American Humanist Association and a bunch of our members and also a bunch of non-members. We had kind of a rainbow coalition of plaintiffs. I think all of the ACLU's plaintiffs were agnostic or atheist. We had a couple of Christians, we had a Wiccan, we had a Jewish rabbi, we had a Christian minister was one of our plaintiffs. So I called it the Rainbow Coalition. And we had talked to the ACLU back and forth about how to do this, and unfortunately we just had irreconcilable differences on the strategy. So we agreed that we would both simultaneously file our lawsuits on May 23rd, 2018. And we showed up at the same time, handed our paperwork over at the same time. Theirs got processed first, so they're the first plaintiffs and we're the consolidated plaintiffs. We got to use their case number, but that's okay. That doesn't mean much because then, of course, the Satanic Temple moved to intervene pretty shortly thereafter, and the state objected strongly to allowing them to join. And their objection was mostly what I'd call character assassination. If you can imagine, how do you assassinate the character of a self-proclaimed Satanist? Now, I thought from a legal standpoint, this objection is ridiculous. But there was some, back and forth about it. And then, finally on December 17th, that's the first big delay, which is like from July 12th to December 17th, the motion to intervene was granted. And the Satanists filed their complaint and intervention on Christmas Eve. Now, there were a lot of comical things that happened during the process of this litigation. I could tell you about the deposition of Rayford. I could tell you about some of the positions taken. I could tell you about the state's discovery that was, comical. But if I did all that, you wouldn't get to hear our speaker today. You might not even get to hear the speaker next time. So I'm going to skip a little bit. One of our points was we thought we should have an expert witness on history because of some recent decisions of the Supreme Court. The ACLU didn't want to do that. So we hired ours. And the Satanic Temple didn't have one, but they had I think they had a different reason for not having a historian. More like their idea was you can't really use a historian. So the Satanic Temple moved to exclude the state's historian, and the state moved to exclude ours. And so we had these arguments. Then we had, and while that was still pending, we pulled the case together so that we could file what's called motions for summary judgment. Summary judgment means that one party or another wins without having to have a trial because the undisputed facts entitle us to a judgment. We said that. ACLU said that, Satanic Temple said that, and the state said that. Everybody had their own motion for summary judgment. And there are a lot of procedural things about those. Again, I'm going to leave that for later. But a hearing was held on that. I don't actually remember the date, but I think it was early July 2023. But nothing happened then for a couple of years. Then on September 30th, 2025, the court issued an order denying all the motions to exclude experts. Now, in most cases, when there is an order to exclude experts that is denied for both sides, that means you're going to trial because that sounds like there's something somebody's going to have to testify about. So I thought, we're getting ready to actually have to go try this thing. I was looking forward to it. But anyway, the court didn't set a trial date. But then on March 31st, at around midnight, actually, the judge issued a 148-page order. The too long didn't read is that we won. And the court directed the Secretary of State, together with his agents, et cetera, to remove the monument. However, the court stayed that because everybody knew this was going to go to the 8th Circuit. So why bother doing it when it's just going to have to wait till the 8th Circuit decides it? So the court said that the removal would be stayed pending a timely appeal to the 8th Circuit, or the time to file an appeal to the 8th Circuit expires, which was, technically that's right, but that wasn't going to happen. Although, as of today, I don't think they've filed one yet, and I would have by now, I can tell you. because a notice of appeal isn't very much. All you got to do is say we're appealing. You don't even have to say why. But this is a big win for us. It is. Because the judge basically, we had different arguments, different approaches, and we won on every one of them. We won. The ACLU won. Satanic Temple won, except one thing is they'd asked to put Baphomet on the state capitol ground for the same period of time that the 11 commandments were there. The judge didn't give them that. But everything else, we won. Now, and that decision is out there. It's, you know, everything that the federal courts do is public. And decisions, you know, should be available to you. And if anybody wants to read it, you can. I'm not really recommending it because I know that as a lawyer, there's a lot of stuff I understand about why this thing is 148 pages instead of 5. And I understand, you know, there's a lot of repetition as you read it. You say, didn't I really read that? But that's because it has to be applied in a different context, so that's correct. But if you want to, it is available. And you can read it, and you can see all the reasoning. You can't really make out the whole case, though, from this order, because in so many cases, the judge just cites to, you know, something else that happened, some other pleading, mostly some exhibit in the record. And, you know, if you don't have that, you can't really quite see what the judge is talking about. As a matter of fact, the first time I really got a chance to fully read this was yesterday. I, you know, read the basics, looked through it, but I thought, you know, I'm going to have to read this with the exhibits available, with other pleadings available, and just check some things, because a lot of times it was just, you know, the reasoning was see, you know, this document, see this exhibit. But I did that yesterday, and it was fun. And I think we're really fortunate that the book club is going to be talking about another constitutional piece of litigation. And a lot of the things that are in there, I can give you some background on, but I think that what we need to remember is that you got to choose your battles wisely, but in the appropriate case, we can have some results. Thank you.
And Jerry, thank you for all of your volunteering and hard work. He was, you know, doing this just, I mean, this is his, I think, capstone of his career. He has made history. He has put his name in the legal records alongside many other lawyers who fought the good fight and have proven again and again and again that the Constitution requires separation of religion and state. So we've made history this month. And you've made history, too, as members of the Arkansas Society of Free Thinkers, because he couldn't have done it alone. FFRF couldn't have done it alone. The AHA couldn't have done it alone. All these great organizations that were able to come together and make this history happen required organization. A lot of the people who were out in the periphery, not involved with organizations, they may have had strong opinions, but it took organization to make it happen. And so when I think about, you know, congratulating Jerry on the hard work, I also have to congratulate you because you showed up, because you paid dues, because you donated, because you helped set up something, because you hosted an event, because you showed up to an event. Just, you know, showing up to events on Meetup helps build this organization. And that from that base, we can launch activism and legal challenges. So if you think that you know, what you're doing isn't important, that's incorrect. It is important. Everything you do contributes to this kind of outcome happening. And so I think we need to encourage a bit more organization and work together on as much as we can. Even if it is just a night going out, it's still something that builds up the organization that our legal team needs to be able to represent you as an organization. Anyway, long-winded. Congratulations, Jerry. And congratulations to the AHA and the FFRF. On Monday, I went through my usual annual routine of going down my list of organizations and donating what I could. I went down and donated to AHA, ACLU, Freedom from Religion Foundation, Americans United, Secular Coalition, Secular Student Alliance, American Atheists, and I didn't have to do anything for Arkansas Society of Freethinkers because I'm on AutoDraft every year. So you can sign up for that too at arfreethinkers.org, and I encourage you to do so. Now for the main event, the show you've all been waiting for, A Tale of Tales Lost by our own Brad Ericson. And let's all give him a warm round of applause and wag your tails in excitement and appreciation.
Okay, now let's see if it's going to work from this distance. I might have to have somebody change the slides because I don't think I'm going to be able to do it from this distance. Is it the big middle button or the top button? Well, that's not doing it either.
Check the remote here and see if it's working at all.
Okay, I think I'm going to get in front here so that I can point some things out from the big screen. But before I get to the actual topic of the day, does anyone know what you're looking at here?
Whale.
A whale, okay, and what part of the whale?
Spout, blow spout.
Yeah, the blowhole. Okay, now a lot of people always think of the blowhole as a hole, but no, it's nostrils. And so at a future date, I'm going to give a talk to this group about the evolution of whales because it is absolutely fascinating. Okay, there we go. Okay, so Tale of Tales Lost. All right, so on the left we have monkeys, and on the right we have apes. What's the most obvious morphological difference between the two groups? Tails, right? So tails are obviously present on monkeys. Okay, so we've got lots of them here. And of course, tails are absent from the apes, including this one. Okay, okay, next please. Okay, so just a reminder of where things are at on the evolutionary tree for primates. Okay, so here's our monkey groups right here. Okay, and then starting with gibbons, we've got all of the various apes. Here's our branch point from our chimpanzee relatives on up to the human lineage. And again, that was about 7 million years ago. Next, please. Okay, so what are the uses for a tail? All right, so tails are adapted to a wide range of environments, and it's primarily determined by the locomotion style of the animal. Now, all assist in balance, but some, like is being depicted here, are used for grasping, and we refer to those as prehensile tails. Note no arms, no. It's the old saying of no hands. You know, so using a tail to get a drink of water. Okay, next please. Okay, so why did the great apes lose their tails? Well, one potential reason okay, was the transition to the ground. Okay, so we went from from arboreal lifestyle to ground locomotion. And a tail actually hinders walking and running. And it also provides no extra stability when you are able to use orthograde motion. So just think of how you typically walk. Okay, you're actually falling as you walk, right? Okay, but because we balance things out, and I'm exaggerating a little here, okay, because we balance things out with right arm, left leg, left arm, right leg, okay, that keeps us stable. Okay, next. Okay, so when did the great apes lose their tails? About 25 million years ago is the point at which this occurred. Okay, so again, all of our monkey species still have them. Again, starting with gibbons, all of the apes do not. And what remains is our little tailbone. And I'm going to define a term here because you'll oftentimes hear creationists misrepresent the tailbone. So what does it mean if we say something is vestigial? Okay, so that is a common definition of it, but does that mean there is no use for it? No, it doesn't. Okay, so what vestigial, the best definition for vestigial is a repurposed use. Okay, so there are attachments, for example, to the coccyx that are useful for sexual reproduction and and other aspects. Okay. Okay, next slide, please. All right, so we're going to have a fair amount of molecular biology today. I'm sorry about that, but there's no way around it. And so I've provided a series of definitions that we'll go through, and then I'm going to provide examples of each of these, and you will need your prop. So does everybody have their prop that wants one? Okay. Okay, so first, transposable elements. Now, Barbara McClintock was the individual that first described these, and she described them in corn. And at that point, she referred to them as jumping genes. Okay, but transposable elements are sequences of DNA that can change their location within the genome. Okay, and there are several ways in which this can take place, and I'll be describing one particular way that's important for our discussion today in a little bit. Then ALU sequences. These are the most abundant transposable elements that are found in the human genome. And again, I'll describe just a little bit more about those here in a little bit. Then exons, introns. Okay, so in eukaryotes, genes are broken up into segments called exons, that is, those portions of the gene that will actually end up being expressed, so the EX, exons. Okay, and they are separated by intervening sequences, so the IN for introns. Okay. And again, we'll take a look at these things in a little bit more detail. Then the process of RNA splicing, that results in the hooking together of the exons and the removal of the introns. And that's necessary for the expression of genes into proteins. Okay, if that doesn't happen, the resulting RNA is not able to be expressed as a protein. Okay, so we have to have that process take place. Then inverted repeats. These are two sequences that are identical or similar in sequence, but they're present in the opposite orientation. Okay, so this would be direct repeats. Okay, one repeat, second repeat. This is inverted repeat. Okay, direct repeat, inverted repeat. And then finally, transcription factors. These are proteins that bind to DNA that promote the expression of a gene by directing RNA polymerase where to begin transcription, that is, the production of the RNA from the DNA gene. Okay, next slide, please. All right, so this illustrates how the ALU sequences are able to move from one portion of the genome to a different portion. Okay, and this process involves what's called a retrotransposon, and it's called retro because we convert an RNA transcript of this from DNA back to a DNA copy, and then it's subsequently integrated at a new position. So that's how this particular element moves from one spot in the genome to a new spot. Okay? Does it through an RNA intermediate. Okay, next please. And so the ALU elements that I'm going to be talking about, this describes a little bit about them. So they're only about 300 base pairs in length, but they're repeated a very large number of times in the human genome, greater than 1 million times. And as a result, you are 10% ALU sequences, okay? Approximately 10% ALU sequence. All right? And they're found mostly in intergenic regions and introns, and they propagate via the process we just discussed, okay, retrotransposition. Okay, next please. Okay, RNA splicing. So this will be the first time we'll use our prop. Okay, so envision this as being the pre-mRNA. Okay, so this is the pre-mRNA, and we've got exons and introns all the way along the length. Okay, now if we're going to get rid of the intron, we have to fold it up and bring 2 exons together. Okay, so, and then if you're really good at handling your licorice, you can get a second one in there, okay? All right, so that's the idea. So we're going to have this folded up, okay, such that the exons are brought together and we cut out the intron sequence, okay? And once that's complete, okay, what we end up with is the spliced mRNA, okay? So all the exons are retained, all the introns are lost. Okay, next please. Okay, so now the other key concept, the inverted repeat. So again, we'll use our prop. Okay, so envision where I've got my fingers here. Okay, so those are regions that are identical or very similar to each other, but they're inverted in reference to each other. Okay, so oops, I just broke my DNA. That's all right, we're good. Okay, so the bottom line to that is we form these stem loop structures. Okay, once we get this converted to single-stranded RNA. Okay, so these sequences are able to base pair with each other, forming this stem loop type structure. Okay, and that'll be very important to why we lost our tails here in just a little bit. Okay, so any questions about any of that? Okay, clear as mud, right? So we're all we're all good. Okay, next please. Okay, so transcription factors. Okay, so transcription, we're only going to concern ourselves with activators. Okay, so transcription factors work by binding to regions in DNA called enhancers. That is, they're going to enhance the expression of a gene. Okay, and the net effect of them binding is to direct RNA polymerase to its proper location to cause gene expression. Okay, next please. And this looks at transcriptional activators in just a little bit more detail. So here's the DNA binding domain, so that's what that DBD stands for. Okay, that binds to the enhancer element. And then this activation domain is what actually interacts with RNA polymerase and directs it to begin transcription. Okay, and again, this will be important in our discussion here in just a little bit. Okay, next please. Okay, so how did they determine what gene was responsible for tail loss? Okay, in other words, what was it that caused that particular gene to no longer function? All right, so by looking at mouse mutants, okay, they were able to identify a candidate gene that results in tail loss in mice. Okay, and this gene is known as TBXT. And this is a transcription factor that's important in early developmental processes. Okay. So experimental examination of the human ortholog, that is, an ortholog is an evolutionarily related gene, revealed some fascinating aspects. Okay, next please. Okay, so the human TBXT gene is found on chromosome 6. So here's chromosome 6. And here's where the TBXT gene is located on chromosome 6. And you'll note that it's comprised of 8 exons and 7 introns. Now, what's very important, what's absolutely key here, is that we have these ALU sequences, ALU SX1 and ALU Y, that flank either side of exon 6. So this is in intron 4, this is in intron 5. Okay? Now note that ALU SX1 proceeds from left to right, whereas ALU Y is arranged proceeding from right to left. Okay, so remember what we described are direct repeats versus inverted repeats. Okay? So let's see how well we're doing. So if we have this sequence, okay, that's highly related to this one, and they're present in the opposite orientation, what is it that we could form once this becomes single-stranded RNA? Sorry? Yeah, a hairpin, okay, our hairpin loop, okay, our stem loop structure, right? Okay. Now, the ALU Y sequence has several characteristics that make it unique. It's hominoid specific, so we only find it in the great apes. Okay, it's present in a gene known for tail formation, okay, for mouse mutants. The proximity and orientation relative to a neighboring ALU element is shared in all simians. Okay, so we find this or arrangement in all of the great apes. And the inferred insertion time corresponds with the loss of tails, okay, about 25 million years ago. So everything's looking like this is going to be what we're after. Yes, Brian?
So those animals that, the ones that do have, monkeys that do have tails, they don't have one or both of those ALU sequences? Or they...
Yeah, so the ALU Y is great ape specific. Okay, we only find that specific element in the great ape lineage.
They have the upstream one, but not the downstream.
Right, right. Okay, next please. All right, so how did they go about investigating whether or not TBXT was the gene, or a gene at least, involved in tail loss in the great apes? All right, so they started by doing some experiments in human embryonic stem cells. So we're going to investigate first the human TBXT gene. Take a look at it. All right, so here's our organization again of TBXT, so 8 exons, 7 introns. And one approach is to directly remove either the ALU Y sequence is being shown here or the ALU SX1 sequence. Okay? And simply ask, do we still lose exon 6 if we remove either of those sequences? Okay, okay, next please. Okay, and the answer is, okay, if we delete one of the ALU sequences, then exon 6 is included. And it's the blue one right there. Okay, so exon 6 is included if we prevent formation of this stem loop structure. Okay, so when the two ALU sequences are present, we form this stem loop structure, and now exon 6 gets treated as if it were intron, gets spliced out, gets removed. Okay. And as proof of concept, if we add an additional ALU sequence that is complementary to ALU SX1, now we can loop out both exon 6 and 7 and create a smaller yet transcript, okay? Create a smaller yet messenger RNA. So this demonstrates that, yeah, okay, this is why we're losing exon 6, okay? So we can make a different construct and delete out 2 exons. Okay, next please. Okay, so what about demonstrating it in animals now? Okay, we demonstrated it in cells. Okay, will this approach work in animals? Okay, so again, here's our TBXT gene, okay, exons 1 through 8. And one approach is to directly delete exon 6. Okay, so if we do that, okay, we have one copy, okay, that contains exon 6. Remember, mice are diploid organisms just like we are. So they're going to have 2 copies of every gene. Okay, so we leave one gene intact. The other, we have removed exon 6. Okay, now if we do that, what do we get? Okay, so here is the mouse that has one copy without exon 6, one copy with. Note that we lose the tail. Okay. Here's the wild type mouse, okay, has the tail. Okay, so no mutation there. All right. And then for comparison, we're looking at the human TBXT, okay, so full length versus delta exon 6. Okay, and we showed that from the previous work in human embryonic stem cells. Okay, next please. Okay, so rather than deleting directly exon 6, what if we put the ALU sequences in there and ask what happens in the whole mouse? Okay, ask what happens then. All right, so here we've placed the sequences in there. Okay, as predicted, we're going to form the stem loop structure. And here's our wild type mice right here. And here's this particular mutant construct. And note that we still get tails, okay, but they are a little bit shorter. Okay, if we put better matched repeats, identical repeats, okay, so remember these aren't identical, they're close, and so the base pairing in this stem structure isn't perfect, okay, but it's good enough to form these structures. Okay, if we put identical repeats in here so that the base pairing is very good, note that we get shorter tails yet. Okay, so these mice have shorter tails. And if we delete exon 6 completely, okay, we once again lose the tails fully. Okay, no tails are produced. So what does this tell us? This tells us that we've got a dosage effect. Okay, so we've got leaky expression, if you will, of 2 copies of full-length TBXT here, because this process is not 100% efficient. This process is better, because we get shorter tails, but we still get some leakage, okay? We still get some TBXT being expressed from that particular allele. Okay? Okay, next, please. Okay, so you might be asking, well, what happens if we delete out exon 6 in both copies? Okay, so if we eliminate exon 6 completely, what happens now? Okay, and to remember our basic genetics a little bit, because we'll need that for the next experiment. So if we take two heterozygotes, okay, so big B, little b for purple flowers, okay, so this is the dominant allele, this is the recessive allele, and we cross it with the same heterozygote, okay, so pistil versus pollen. Okay, note that anytime we've got the dominant allele, we get purple flowers. Okay, when we have the double recessive, okay, we get white flowers. All right? And note the ratio also. So we've got this being a fully dominant homozygous individual, this being a fully recessive homozygous individual, and we get two heterozygotes, so a 1/4, 1/2, 1/4 arrangement, okay, when we cross heterozygotes. Okay, next, please. Okay, so how do they go about generating mice that have the exon 6 fully deleted? And it was done as follows. So they crossed heterozygous mice. Okay, and so here's our delta TBXT exon 6 wild type crossed with the same same genotype. And again, we'll yield 1/4 wild type, so wild type plus wild type giving us this. Okay, and 1/2 heterozygotes, so this with this and this with that gives us our 1/2. And then finally, 1/4, okay, are fully recessive for the TBXT gene. Okay, the net result of that experiment is that the delta TBXT E6 mutation was lethal. Okay, some mice were arrested during development, others were born, but quickly died of spinal cord defects. Okay, so in other words, their development was stopped before being born. Okay, okay, so yeah, could be stillborn, could be even earlier. Yeah. Okay, next, please. Okay, so conclusions. So a plausible evolutionary scenario for tail loss in hominoids was due to the insertion of an ALU sequence element into the TBXT gene. Okay, the net result of this insertion is to cause exon 6 of the TBXT gene to be skipped, producing a mutant form of the transcript. Experimental deletion of either ALU elements prevents exon 6 loss in embryonic stem cells. Mice engineered to remove exon 6 or have inverted repeats inserted into introns 5 and 6 resulted in partial or total loss of their tails. The peptide sequence of exon 6 encodes a partial transcription activation domain but does not influence DNA binding. Okay, so this mutant form still binds to DNA, still binds to the enhancer, but it's the exon 6 component that directs the binding of RNA polymerase. Okay, that's the activation domain. Okay, next, please. Okay, so therefore this may alter transcription activation functions of TBXT. Our hominoid ancestors lost their tails, at least in part, due to the insertion of ALU-Y approximately 25 million years ago. And additional genetic changes have probably stabilized the tail loss phenotype. Thus, removal of the ALU-Y is unlikely to cause tail formation. Okay. Now, every once in a while, you do see an extended coccyx in humans. Okay. And so why might that be? Well, remember I said that we get some leakage, okay, because we don't form perfect stem loop structures. Okay, so if they would happen to have a mutation that might accentuate that leakage, okay, that could result in that formation. Now, evolutionary pressures for tail loss are probably locomotion related, and the pressures must be great as we have an evolutionary trade-off of neural tube defects as evidenced by mice lacking TBXT. I think everyone's heard of spina bifida, okay? So that's one of the maladies that maps to this particular gene in humans, okay? And mutations leading to neural tube defects have been detected in both the coding and non-coding regions of TBXT. Okay, next. Thanks very much. You may eat your DNA if you haven't done so.
So when we talk about the evolutionary pressure that might have led to this, and where 25 million years ago apes lost their tails and started living at least part of their life on the back legs, I'm wondering if the tailbone has an important role sort of holding the guts in, so to speak, when you go from being a horizontal four-legged animal to a vertically inclined two-legged animal, and the pressure is now down on your pelvic floor, especially during pregnancy maybe. Is that maybe a hypothetical source of that pressure? Is that suddenly you take a four-legged animal, you stand them up, and the fetus is not as well supported?
Perhaps. There's additional mutations, and in fact, it's amazing that it's really only 2 mutations that are responsible for reorientating the pelvis for standing upright. And so we would have to also have, okay, additional mutations that would allow for that. Okay, now, wish we had Gutsick Gibbon here because this is her area of expertise. But anyway, yeah, I mean, that could be part of it, part of the story. Yep. Yes? Go ahead.
Is it known why having both copies of exon 6 deleted was lethal to the mice?
Yes, so let's see. Let me jump back real quick. Okay, keep going, keep going. A little bit farther, keep going. We'll get there. There, okay? So if we lose both copies, okay, what does that mean? That means that whatever genes are being, whatever genes expression are being driven by TBXT binding, all of them will be shut off, okay? So TBXT isn't just responsible for the expression of one gene, okay? It's a transcription factor, so whatever genes happen to have that DNA binding domain sequence, okay, that transcription factor can recognize, those would all be off. Okay, so that's one. Yes, Brian.
So I missed something about the picture of the mice with shorter and shorter tails. And the last couple of mice you showed, what was deleted in that one? Was it both?
Right. So there. Yeah. So let's again move through the slides. Sorry, Chris. Okay, find the one where we show the mice with their tails. There, okay. All right, so note that what we've done in the final panel, the right-hand panel, is we've deleted exon 6 completely. We just directly cut it out. Okay? So we're not dependent upon formation of the stem loop structures, okay, to eliminate exon 6 anymore. It's already gone. Okay, we engineered that portion of the gene directly out. Okay, so what that's telling us, okay, again, is that the formation of those stem loop structures, followed by the removal of exon 6, treating it as intron, is not fully efficient. Okay? We still get some expression from that copy even though we form stem loop structures. Okay?
So the last two mice, they have exon 6 completely removed from both copies?
No, just
That's
one.
what I didn't understand.
Just one copy.
Okay.
Yeah, just one. Yeah, again, if we did it in both copies, that's lethal. Yeah. Yes. Yeah.
Nature didn't do that. And when we come back to the monkey and the ape, one travels on the walking, and the monkey with the tail travels in the trees.
Right.
So there's the reason that the ape who had lost the tail didn't need the tail anymore, because it started walking on the ground. And the monkeys retained their tail because they continued to swing in the trees. I'm sorry, I have a loud voice. Is that it? Is that why?
Well, that's certainly a driving force that would be selected for. Okay, so if you're going to have, like I said, orthograde movement, okay, a tail will interfere with that type of movement.
Yes.
Okay, and it does not provide any extra stability by having a tail for that type of movement.
Their survivability would be less, and they would breed less, and they would be.
That's right. That's right. So you're not going to be able to run as fast or walk as fast, however you want to think of it, if you have a tail.
Catch your monkey by the tail.
That's right.
Wow.
So, fascinating tale, and it's really quite amazing. And just as kind of a prediction, I think eventually we will know gene by gene what changes took place to generate where we are, okay, from our evolutionary history. We've seen a bunch of this happening over and over again. This is one example. Our larger brains is another example. We know at least one gene that's very important for that. Like I said, we have identified 2 genes that are responsible for changing the hip orientation for being able to walk upright. So it's happening.
I think what's more, I call more perplexing or more, maybe out there and stuff like that, but maybe if it comes to light that, disclosure movement, all these things people see they can't explain, evidence we all world's extraterrestrial, more intelligent beings may have had a hand in our development, that's our DNA. And if that is disclosed, all these classified files, that maybe it's possible that we were actually somewhere else being messed with our development, or not. Or, I know it's out there, but there's a lot of stuff there maybe revealed, not their awareness of people, beings that are far ahead of us, evolved, not Earth, all worlds, whatever you call them. And that conspiracy theory, but I think it's gonna have to have it reality.
Right?
So are you saying that we do or don't know the details about why we get by without both copies and manage to compensate, not die, or because both copies are missing? Do you know about that or don't?
Yeah, so if we end up with both copies being defective, okay, or in other words, if our TBXT dosage drops quite a ways, okay, then that's when you see those neural tube defects, stuff like that developing in humans. And as far as I know, I doubt you could get a human to parturition without at least some functional TBXT. Yes?
Don't we have larger brains because of better nutrition? That's why we literally have the bigger brains. And I noticed that E.O. Wilson in his book Conciliums said that we cannot grow any larger brain. The cranium will not open up any more, stretch, and, pieces will not stretch any more than they are now.
Well, going to a protein-heavy diet, and maybe that's what you're referring to, was definitely important in allowing larger brain development. But there is an experiment done not too long ago. I forget the group that did it, but they had identified a particular gene that was responsible, or at least what they thought was going to be responsible for generating larger brains. And they placed that gene into, I think it was rhesus monkeys, and voila, they were starting to develop larger brains. They terminated the experiment because of ethical reasons. Imagine a bigger brained rhesus monkey. How are you going to treat that individual? Would it, just how far would it go? Would it, so anyway, they terminated that experiment because of ethical reasons. They were concerned that, maybe it would be at the level of a, highly mentally retarded human, or, they didn't know exactly what they would have gotten. But they do know that it did increase the brain size.
So what happens to the bone structure of the cranium? Because that's
Right, now remember that the cranium is actually rather pliable, until several years after birth, actually. And so I don't see that as a big problem, but not to destroy the Adam and Eve story, but why do women experience childbirth pain? Why is that? Well, it's because we evolved larger craniums, okay, quicker than we've evolved larger pelvises. Okay, and so as a result, women experience that childbirth pain, whereas basically no other mammals have that issue. They do not do that. They give birth very easily. So.
Who did they ask?
I think they also showed that intelligence, like creativity, imagination, like people that have such invention, like people have great music, such extraordinary works, creation, they have the same size brain, and their mind is the same size as anyone else, yet they can create magnificent machinery, like all these inventions, cool things. That's
Sure.
Imagination. But I think they also discovered like psychological trauma. Mental trauma can retard and reduce your intelligence, or self-doubt, or if you're raised in an environment where you're not encouraged to make you feel like you're not as smart, you're less stable, like false belief that you're not as bright as other people,
Mm-hmm.
Plus, it's all like resources. People are very intelligent, where they have access to develop their natural skills. They're not people that nourish them and encourage, they're help them create their own inner creativity.
Yep. Nature versus nurture, right? So, but yeah, with music ability, I can envision a way in which you could try, and maybe somebody's done this. It could be, but I can envision very specific tests, like you might test somebody for perfect pitch. Individuals that have perfect pitch, you might try to track down the gene or genes that are involved in someone having that, and be able to identify what would be necessary to be a great musician.
Right, quality trait stuff.
Mhm.
Yeah.
Yeah. Yes.
I'm a Jamesian, and Dr. James, in his Origins of Consciousness and the Breakdown of the Bicameral Mind, rather proves that back in Egypt, the people
I mean, by the time of the Egyptians, though, I think we are pretty much the same brain size now as they were then. I'm not aware of any expansion of the brain case for that point.
Well, one example he uses in there is Homer's Iliad and Odyssey from the Greeks, in which, if you notice in the book, the people don't do any thinking for themselves. All is done by God telling them, or their imaginary God in their head, telling them to go forth or pull back, and so on, in those books. So the same thing, I mean, Egypt goes back a long time before Cleopatra, I mean, way back.
Sure, but I mean, Homo sapiens as a species has been around at least 200,000, and some people say around 300,000 years. So that's obviously a lot longer than that. So.
Just a minor point about perfect pitch. I read something several months ago, I don't remember the detail, but it implied that perfect pitch could actually be taught. I'm not sure that everyone could learn to have perfect pitch, but it implied that at least some individuals were trained to have perfect pitch. I think it said as children, not as adults.
Right.
All the details.
I mean, music I could see being similar to language. So I mean, you may very well be able to do that.
I'm the opposite end of perfect pitch. I can't tell anything.
I suspect that perfect pitch is going to be more a matter of changes in the inner ear than they are the change of the brain. It's just a mechanical process of processing sound. And the other thought is that just making a bigger brain does not necessarily make you smarter. The brain has to be organized enough to actually do something.
Sure.
That's what I allude to, yeah, the science.
Sure. No, it's very clear that some people have completely lost one hemisphere, and yet get along just fine because the brain is able to rewire itself in a manner that no one understands yet. Okay,
They're not perfect, they can't do a 8-hour job like that.
Right. Yeah.
So, I just want to say to the group, UU Book Club is now reading a book called Music Cophilia by Saxon. And this is all about music and how, it plays in people's heads and so on when they get different illnesses, diseases, and so on. It changes what happens to the way they hear music. If anybody's interested, just want to mention that book. It's a fairly interesting book about neurology and
Recurring
music.
tunes, so that's what recurring tunes,
That's what, yeah.
They get in your head like tingles, they know the programming.
On the brain science, there have been apparently numerous studies that show that there was a correlation between intelligence and not brain size directly, but the brain size to body size. So, for example, elephants have a much larger brain than us, but relative to body size, it's
Yes.
smaller. And they have a lower, they're smart, but they're not as smart as us. But their brain to body size ratio is less than ours.
You were talking about perfect pitch. I'm in the quartet. The lead and the tenor of my quartet says you can learn it, but I'm not a good candidate. Anybody else?
All right. Well, thanks so very much.
Thank you, sir. Thank you.
Thanks everybody for stopping by today, and again, check out arfreethinkers.org for either a calendar of events, a way to join ASF, or information about the group. Our best source for information about meetups is on meetup.com/arfreethinkers. And we've got several events coming up this month, actually, and there's a sign in the back that describes them as well. We've got Pint Night happening tomorrow night at 7 at the Flying Saucer. Then we have a board meeting on the 21st if you want to get involved in ASF leadership or just hear what we're up to, feel free to drop in on that. Links in the meetup. And on the Thursdays, we have Beyond Belief, Secular Substance Abuse Recovery. Next Sunday, so a week from now, at 2 p.m., we have the ASF Book Club. We're reading The Devil in Dover, the story of a intelligent design court case in Pennsylvania, I believe it was. And so we'll be meeting up next week to discuss that at the filling station on JFK in Little Rock, North Little Rock, I mean. And then looking forward past that, on Monday the 27th, we've got Fourth Monday Discord night. If you're not on Discord, that's a great way to meet up and have a get-together and discussion without even leaving the comforts of home. And May the 5th is the first Tuesday of the month of May. And of course, we have an event called
First Tuesday.
All right, Winter Winter Chicken Dinner. And first Tuesday is at American Pie Pizza in the up off of McCain and North Hills in that area. And then, of course, after that, there is going to be Second Sunday Cantina Communion on the 10th of May. And that is at Cantina Cinco de Mayo on Stagecoach Road.
What time would that be?
I'm blanking. What
6
time is it?
o'clock.
6 o'clock. Another winner. Good job. And so anyways, you can keep up with ASF events using the calendar on meetup.com, and you can donate directly. You can become a member. You can get involved in a number of ways. We need help with whatever skill you have, whether you can work technology, whether you can write, whether you have skills in the legal field, the business field, if you want to come up and give a presentation, the month of June is open. Next month, we have a speaker from Better Angels, which is an organization devoted to helping people bridge the communication gaps between political ideologies and keep things rational, friendly, and really right up our alley. So I'm really looking forward to that. Encourage everybody to come out next month for our speaker. But June is open, so if you know anybody, send them our way, or if you are that somebody, talk to me about becoming a speaker, and you don't have to do as good a job as Brad did, but, you know, good enough is good enough. Thank you for everyone for coming, and I will see you all next time.